Okay, so in this video, I'm just going to try and explain the whole of respiration. I'm going to be talking about glycolysis, link reaction, Krebs cycle. I'm also going to do a bit of anaerobic respiration as well, and then finally I'm going to explain oxidative phosphorylation. So, simply, I'm just going to start with the concept of respiration. From GCSE, you'll know that respiration, the equation for respiration is C6H12O6 plus 6 molecules of oxygen goes to make 6 carbon dioxide plus 6H2O. This is the general equation for respiration. So quite simply, at GCSE, all you need to know was 1 molecule of glucose over here and 6 molecules of O2. They are the reactants in this equation, and the products here are carbon dioxide and water. And I'm going to go into detail about how this is true and where this is used. But quite simply, this is the equation for respiration, and yeah, as I said, I'll explain this. So the first step is glycolysis. So as I just showed, I'm going to put this back. As I just showed, glucose is one of the reactants, so I'm going to show you how now. So we're going to start with a glucose molecule. So obviously this is C6H12O6. I'm also going to put the numbers of carbons here. So we start with glucose. By the way, this is called glycolysis, what I'm just going to say. So yeah, glycolysis. So glycolysis, and what glycolysis basically means is the breakdown of glucose into a molecule of what we call pyruvate, and this is going to release energy. So yeah, I'm just going to go through glycolysis. So we have a molecule of glucose. This gets broken down into a molecule of 1,6-fructose bisphosphate. Again, apologies for the handwriting. I'm also going to move this across to make it more central. Again, yeah, I would suggest speeding this video up because I tend to make some mistakes and I have to correct them. So yeah. 1,6-fructose bisphosphate, and this is also a 6-carbon chain. Now, this reaction occurs because we use 2 molecules of ATP, and we form. Maybe I should connect that. And we form 2 molecules of ADP plus a phosphate group, plus 2 phosphate groups. Obviously, maybe I should put some brackets instead. 2 ATPs and phosphate group. So you can see that ATP here is being hydrolyzed to produce 2 phosphate groups. And the phosphate groups here attach to this molecule here, fructose bisphosphate, hence the bisphosphate. Bisphosphate means 2 phosphate groups, and obviously we're producing 2 phosphate groups here. This is a very unstable molecule, 1,6-fructose bisphosphate. So it breaks down. I'm going to do this 2-way, and I'll explain why. So it breaks down into a molecule called triose phosphate. I'm going to make this shorter. Apologies. Triose phosphate, and what you'll notice, triose, suggesting that it's a 3-carbon compound, and you'd be correct. But this splits into 2 3-carbon compounds because we've got 6 carbons and we're producing 2 molecules of triphosphate. I'm going to draw this as like 2 separate pathways because it's much easier, and it's much easier to visualize as well. This happens conventionally because this is a very unstable molecule. 1,6-fructose bisphosphate is very unstable, so it breaks down into triphosphate. Now, this triphosphate is also unstable, but it requires the conversion of substances to produce a compound called pyruvate, which is also a 3-carbon compound. Pyruvate is a 3-carbon compound, and obviously same here. Pyruvate, 3 carbons. Okay, and this requires adenosine diphosphate plus phosphate groups, and this requires 2 of them to produce 2 molecules of ATP, and of course this happens on this side as well. So it's very difficult to write with this pen. 2 molecules of ATP, but also we're going to produce a molecule called reduced NAD. The way we do that is through reducing the molecule of NAD. This should be plus. So NAD and reduced NAD, they are coenzymes. So NAD plus those 2 make NAD H. Okay, you'll notice that overall in glycolysis, we used up 2 ATP. So right now we start at net zero gain of anything. Now we've lost 2 ATP, so we've used energy in glycolysis. But then when you form pyruvate, you'll notice that per molecule of triphosphate, we also produce 2 ATP. However, we have 2 molecules of triphosphate, so net gain over here would be 2 ATP plus 2 ATP minus 2 ATP because we've used ATP here. So we've actually, from glycolysis, have gained a net of 2 ATP molecules, and we've also produced 2 molecules of NADH here, NADH, reduced NAD, because obviously per glucose molecule, we're producing 2 molecules of triphosphate and 2 molecules of pyruvate. So the idea here is per molecule of glucose, we have in glycolysis gained 2 molecules of ATP and 2 molecules of reduced NAD, which are coenzymes. Okay, now I'm going to keep this page up, but now I'm going to go into something called the link reaction. Link reaction. Okay, what the link reaction is basically a little intermediate step between the between glycolysis and Krebs cycle. And the reason we have the link reaction is to produce a product that can be used in the Krebs cycle, and this is called acetyl coenzyme A. So you'll see from glycolysis, we've produced a molecule of pyruvate, or 2 molecules per glucose molecule, but we'll just use one here to make it clear. Remember, you're going to produce 2 molecules of pyruvate, so both of these molecules are going to undergo this. So pyruvate's obviously 3 carbons, and all in all, you'll produce a molecule called acetyl coenzyme A, but I'm just going to call it CoA to make it easier. However, interestingly, this is actually a 2-carbon compound. So this suggests that you've lost a carbon, and this is true because you've lost CO2 here. But also, for this reaction to take place, we also have a molecule of NAD+, another one, that gets reduced into reduced NAD. And also to form acetyl CoA, you're going to have to add an enzyme, a protein called, a coenzyme called coenzyme A. I'm just going to call it CoA, but remember this is coenzyme A in the exams. I suggest writing them as coenzyme. So yeah, so this is a link reaction. So per molecule of pyruvate, we've released a molecule of CO2, which is actually one of our products. It's a waste product in this instance, but from the equation that I showed at the beginning of the video, this is one of the products that you form, and this is how it's formed. So carbon dioxide leaves, so make this a 2-carbon compound. You've used NAD to reduce NAD. So obviously you've got 2 molecules of pyruvate, so you're going to produce 2 NADs, reduced NADs, and obviously the addition of coenzyme A allows the pyruvate to form acetyl coenzyme A. So that's a link reaction. And the importance of the link reaction is just to produce this compound here, acetyl CoA, and I'll explain in the next step why this is important. I'm just going to add another page here. Okay, so now I'm going to move on to something called the Krebs cycle, which is going to use this acetyl CoA that we just used. So the first thing that we need to do is draw our product of basically the cycle. So we're going to start with a 4-carbon compound called oxaloacetate. I'm going to make this smaller because it's quite a big diagram to draw. Hopefully that's big enough. Here it's fine. And this is obviously a 4-carbon compound chain, and this is going to produce a molecule of citrate, which is a 6-carbon compound. You'll wonder where the 2-carbon compound, 2 carbons have come from, and this has come from the acetyl CoA, which is over here, acetyl coenzyme A, and this also releases coenzyme A, which can then, of course, be used again here in the production of acetyl CoA. So this can happen again and again. So oxaloacetate uses acetyl CoA, uses 2 carbons to form citrate. You also do have an intermediate compound here, a 5-carbon one, but you don't need to know that. You can just go straight here. And basically, for this cycle to occur, we need, so obviously, you can see that we've gone from a 6-carbon compound to a 4-carbon compound. So we're going to release 2 molecules of CO2, which again contributes to the overall equation because we've got CO2. It's a product, it's a waste product here. And we're also going to use more NAD, NAD+, excuse me. And we're going to produce, apologies, I made a mistake. We're going to use 3 molecules of NAD plus to form 3 lots of NAD H. And also, we're going to produce another molecule of ATP through condensation reaction of adenosine diphosphate plus a phosphate group. And finally, we also have another coenzyme called FAD. This is plus, which is going to form reduced FAD, which is represented by FAD H2. So that's Krebs. So you can see overall, we started with acetyl coenzyme A. We released that acetyl coenzyme A to bind with oxaloacetate, which is a 4-carbon compound, to produce a 6-carbon compound called citrate. And to produce citrate to form oxaloacetate, we lose 2 carbon dioxides, produce 3 lots of reduced NAD, 1 molecule of reduced FAD, and we also produce a molecule of ATP. So before I go on to oxidative phosphorylation, I think I'm just going to explain anaerobic respiration quickly. It shouldn't be too difficult. So anaerobic respiration, what I'm going to do for this one is refer back to glycolysis here. I'm actually just going to copy all of this. Copy, and I'm going to paste it into this new page. Paste that here. Okay, cool. So, yeah. So specifically, I'm going to look at the conversion from triphosphate to pyruvate here. This is what's important in anaerobic respiration. So in mammals, pyruvate is removed by converting it into a molecule of lactate, and I'm going to show you how. So I'm just going to remove this top part, and we're going to specifically focus on triphosphate conversion to pyruvate. I only need one of these cycles, so I'm going to get rid of this one. Yeah, I should probably copy that one itself, but that's okay. I'm gonna move the center. It's a little bit bigger. Okay, so you can see the cycle remains here as triphosphate forms pyruvate. We produce a molecule of reduced NAD, and we produce 2 molecules of ATP. Now, what's important, specifically to form lactate, is the NADs here. So I'm actually going to switch the sides because it's going to be more convenient to draw on the right side. So, as I said, NAD plus forms NADH. Obviously, we do also produce ATP still, but I won't do that. So, what you need to know, I'm also going to remove this 3 carbon. What you need to know is that pyruvate forms a molecule of lactate. And this reaction here requires reduced NAD for it to take place. So, just really quickly, I'm going to remove this here to make it more convenient for this diagram. So, we use reduced NAD, and we're going to produce NAD. This kind of acts like a catalyst, or an enzyme in this case. And although it's not a protein, actually it is a protein, it's a coenzyme. So yeah, this acts as an enzyme here. NAD plus, which can then actually be used in the cycle again to produce NADH. So this cycle keeps happening to form lactate. And of course, in this process, you keep on forming, remember that we use ADP, and you keep on forming 2 lots of ATP. So this reaction itself is actually just producing ATP in itself. However, because you're producing lactate, lactate's not as safe in the blood as well. So you gather what's known as oxygen debt, or I think excess post-exercise oxygen, an EPOC. So when you're exercise, or when you stop producing lactate, oxygen is available again. So the pyruvate in the liver is basically, it gets oxidized through this link reaction, through the link reaction below, or above rather. And yeah, so the pyruvate can then be converted into glucose and whatnot. So yeah, so that's lactate fermentation. We can also move on to ethanol fermentation really quickly before I move on to the big boy, oxidative phosphorylation. So, triose phosphate, 3 carbons, forms pyruvate, and this obviously produces. Sorry, I don't know what I'm doing. This produces 2 ADP, which is going to form 2 ATP. Okay, so in ethanol fermentation, you're going to overall produce ethanol, but between that, you're going to produce ethanol, which is then converted into ethanol. Again, I'm going to make this shorter. I think we're about 20 minutes in, so I would highly recommend speeding this video up because I'm a very slow talker. Anyways, so you produce ethanol, and if you take chemistry, you'll obviously realize that these two are 2 carbons, and this is 3 carbons. So you're obviously here you're removing CO2. Yeah. So the conversion from ethanol to ethanol is catalyzed by the enzyme ethanol dehydrogenase. Yeah, and it also requires, like similar to lactate fermentation, it requires NADH, which is obviously a product of this reaction here, triphosphate to pyruvate, and it's going to produce NAD because it's chemically changed now. However, then it comes back and forms NADH. So again, this cycle is repeated and repeated, and you're just producing ATP as it's repeated because you form NAD, and that's going to form NAD plus through glycolysis of triphosphate to pyruvate, which allows this reaction to happen, produce a lot of ethanol, which is also quite toxic. So it does need to be removed, and obviously the organism would much rather aerobically respire. But anaerobic respiration basically just allows ATP to be formed. Okay, so that's anaerobic respiration out of the way. So now we can move on to the relatively difficult topic, which is oxidative phosphorylation. Okay, so to understand oxidative phosphorylation, I'm going to draw a mitochondria. I'm just going to label them. So this is obviously the mitochondria. I'm going to excuse the atrocious drawing. Okay, so what I've drawn, or what I've attempted to draw here, is the mitochondria. So I'm going to start labeling this. So this is obviously, this is obviously the outer membrane. This is going to be the inner membrane. And the space in between the outer and inner membrane is known as the intermembranal space. And you'll see that I've drawn like inner foldings. These are called cristae, and basically these cristae allow for high surface area. So more protein molecules can fit into the membrane to allow for this oxidative phosphorylation to happen at a high rate, because you can fit more proteins in. And also, the space here in between the inner membranes is called the matrix. It's like the cytoplasm of an animal cell almost. Yeah, and it's also important to remember that this is obviously a prokaryotic cell. So yeah, so I briefly mentioned that on the inner membrane you have like small proteins, and I'll explain what those proteins are in a second. So yeah, so this is the membrane. You just need to remember intermembranal space, outer membrane here, inner membrane, cristae for large surface area, and matrix. And on the surface of the cristae you have proteins, which I'll explain in a second. So yeah, so this is important to remember in oxidative phosphorylation. So I'm going to zoom in into the membrane here. Not literally, I'm just literally going to draw it out. So I've drawn phospholipid bilayer every time. Actually, yeah, I'll just draw the phospholipid bilayer. Actually, I won't use green. I'll use something else. Convenient. Yeah, let's use blue. So yeah, again, speeding this up is highly recommended. So I'm gonna paste that. Yeah, so I suppose this is efficient. Sorry. So actually I'm going to delete this. Oh my god. So we have what I've drawn here is the inner membrane. Actually, it's going to be more convenient for me to draw just them as lines because you know they're phospholipid bilayer anyway. But it's important to remember that they are phospholipid bilayer for one reason that I'll just explain in a moment. So we have the inner foldings of the cristae, obviously, and we have the normal outer membrane as well. And this is obviously the inner membranal space. So on the surface of the apologies, I'm going to draw this the other way. So this is going to be the matrix side, and this is going to be the inner membrane. Obviously, you have cristae here, but for the sake of it, I'm not going to draw that. And then you have the outer membrane here. Yeah, so this is outer the mitochondria. And obviously, this space here between here is known as the intermembranal space. Intermembranal space. Okay, and basically, what you need to know is that you have electron carrier cycle known as the electron transport chain. And yeah, so basically, we've seen that in the Krebs cycle, we produce 3 molecules of reduced NAD and also a molecule of reduced FAD. And why this is important is because reduced NAD comes along and donates one of its electrons to the electron transport chain to produce NAD. And FAD also does the same. Reduced FAD also does the same. So all of a sudden, we have electrons here in the electron transport chain. And as you know from the photosynthesis topic, when electrons move down the electron transport chain, they lose energy, and energy is released. In the matrix, we also have H plus roaming around. And the energy produced from the electron transport chain, or the electrons from the electron transport chain, moving down them, allows the H plus ions to go into the intermembranal space. So all of a sudden, you have loads and loads of H plus ions. And the reason that H plus can't just go into the intermembranal space, although it's in high concentration here in the matrix, is because of the phospholipid bilayer. It's impermeable to H plus ions because H plus ion is an inorganic ion and is water soluble, so it cannot pass through the subsurface membrane or the inner membrane of the mitochondria. So it needs to pass through proteins here, and they're pumped through a protein channel by active transport. So yeah, you have H plus here, just running around, and electrons are obviously moving down the electron transport chain, which is producing more and more energy. However, now you have a buildup of H plus ions here in the intermembranal space. So you also have what's known as a protein ATP synthase molecule, which also has a channel here for H plus to go through. And as H plus moves through the ATP synthase molecule, this is going to produce ATP because adenosine diphosphate is phosphorylized through the condensation reaction and produces ATP. So now H plus, H plus. And the reason that this mechanism is called oxidative phosphorylation is because you also have oxygen involved at the end. So when electrons have used all their energy and produce all their energy in the electron transport chain, they are accepted by oxygen molecules, which also bind to H plus to form water. And I'm going to balance this now. So 2H plus to this, half of this produces a molecule of H2O. So oxygen here is known as the final electron acceptor because it accepts the electrons at the end of the electron transport chain. Hence why it's known as oxidative, because oxygen is accepting the electrons, and phosphorylation because adenosine diphosphate is phosphorylized to produce adenosine triphosphate. So yeah, I think that's it. I can't think of anything else. Yeah. So to quickly summarize, oh yeah, I'll just mention quickly. Obviously oxygen, you've used oxygen and you're producing water. So to reiterate, in glycolysis we used a molecule of glucose, and over here we're going to use another reactant of oxygen. So obviously 6 molecules, and that produces 6 molecules of CO2. We produce CO2 in both Krebs cycle here, CO2, and we also produce CO2 in the link reaction here. So we produce CO2, and we also have now produced H2O here. So yeah, that's how respiration works. I think I covered everything. I've done aerobic here, oxidative phosphorylation, and anaerobic respiration. So to quickly summarize, we start by glycolysis, which produces 2 molecules of pyruvate. Pyruvate then forms acetyl-coenzyme A, which is needed in the Krebs cycle, and we also form reduced NAD. In the glycolysis, by the way, per molecule of glucose we have a net gain of 2 adenosine triphosphate and 2 molecules of reduced NAD. In Krebs cycle, we produce more reduced NAD, and we also produce reduced FAD, and we also produce ATP. We release carbon dioxide, and I did also mention anaerobic respiration of ethanol fermentation and lactate fermentation. And in the mitochondria, we have the matrix intermembranous space, in which you have an ATP synthase molecule which allows facilitated diffusion of hydrogen plus, which reduces ATP, and oxygen is the final acceptor at the end of the electron transport chain. Hence, it is known as the final electron acceptor, which reduces H2O, and hence is called oxidative phosphorylation. So yeah, I think that's all. If you have any questions, please do feel free to ask. Yeah, because it's quite a tough topic. It took me a while to understand as well. So yeah, I think that's it. All right.